Hey guys, so this is the video where we're going to go over what we saw in our plant structure and vascular tissue lab. Okay, this was the first lab that we did, actually the second one after we made our cells made our slides from chick cells. Okay, so this is the second lab where we actually had all the slides and some of the plants under the microscopes, and we looked at some stuff and we label different parts on it. So here we're looking at the roots. Okay, this is through the dissecting microscope, and you can see I already labeled the parts. The root hairs are the little branches that are coming off the root. You can see them there where I label them. So that's the root hairs. Remember, root hairs are for to maximize the absorption of water for the plant, okay, and then I label the root. So that's the main branch. That's your root. So if I show you this in your practical and your test, make sure you're able to identify the root hairs and the roots, okay. We also looked at stems, and we said that stems, their main function is to support the plant, okay, support the branches that hold the leaves, that hold the flowers. So again, their main function is to support the plant, to give it a structure. They also have stem hairs, okay, they also have hair, and that hair is not to maximize the absorption of nutrients, it's actually to protect the plant from predators or herbivores, things that are going to be eating the plant, okay. In some plants, the stem, not the stem hair, the stem actually absorbs nutrients and it expands, like cactus, right? Cactus, they expand, their stem expands, so they store nutrients, they store water in there. Okay, so that's stems. We also looked at celery, and we dipped the celery in water with red food coloring, and that's why the xylem, we're pointing to the red area in the picture as the xylem, and the phloem right underneath it. Remember, xylem absorbs or moves water up to the leaves from the roots. Phloem moves glucose and nutrients all over the plant, right? So xylem flows up, phloem flows in multiple directions, okay? So again, the reason the red part is xylem is because the water gets absorbed up the stems, in this case the celery, and since we dipped it in water that was colored red, then that's going to show us the xylem, okay? This is station 3, where you guys looked at a leaf under the dissecting microscope. And all I want you to see here is the shiny stuff on top of the leaf. You can see that that's called the cuticle. It looks like wax on top of it. Okay, and the cuticle is there to prevent water loss. This is one of the main adaptations that plants have developed in order to survive in different environments. So that's what I wanted you to be looking at, and that's that waxy covering on top of the leaves, and that's called the cuticle, and is there to prevent water loss. Okay. Now this is the cross section of a leaf, and I wanted you guys to label the xylem and the phloem. I already labeled those, as well as the guard cells and the stomata. But if you look at here, I'll go ahead and add, let me grab another color real quick. This, okay, is called a vascular bundle. So when we're looking at xylem and phloem together, we call those vascular bundles. That's one bundle, this is another bundle, okay? And you can see that all these little circles here, okay, they're empty, right? And those are your xylem. Okay, remember that xylem is a non-living cell or a non-living thing, okay? Phloem is a living cell, and that's this big ones out here, okay? If we had other microscopes, sometimes we'll be able to see some type of structures inside the cell. They're living cells, okay? Xylem is not. So the other part that I wanted you to label was the stomata opening and the guard cells. If you look at these cells down here, all of them are touching, all of them are connecting, okay? Except these two, right? So these two actually have some type of opening here. And that's what the stomata opening, that's where the stomata is, right? So these openings right here are called stomata, and what they do is they open and close to release or exchange gases with the outside environment, oxygen and carbon dioxide, right? Water leaves through there as well. And the ones responsible for closing and opening the stomata are called the guard cells. That's the two cells right there that are next to the opening, okay? So make sure you're able to identify these parts: xylem, phloem, guard cells, and the stomata. Make sure you know the function of these, and if I show you this slide in a microscope, make sure you're able to identify these parts. Okay. All right, the next one was the underside of the leaf, and this is how stomata looks when you have the underside of a leaf. And if we look at this one closer, right, we see a kind of like a donut or circular shape, and then we see the opening here. And around there you have these cells that look like that, okay? So if we use this one, that would be the stomata, okay? And these two cells would be the guard cells, okay? Just like I labeled it up there, I'm just going over another one of the stomata on there. So make sure you're able to identify that. Remember that a lot of the stomata is going to be on the underside of the leaf because the sun doesn't hit it directly, so it's much cooler, and not a lot of water is going to be lost. Okay, so there's a lot of stomata on the top. If you compare the underside, you can see all kinds of stomata here. If you go look at the top side, there's no stomata on this leaf at least. The only thing we could see is the cuticle, which we already mentioned earlier, and that's just to prevent water loss. Okay, this is a root that you guys looked at. Okay. And you can see all the phloem, all these circular empty circles here are phloem. Okay, and then the little structures or the cells around it, which is usually up here, this area, okay, those are your phloem. Okay, now we know that if we circle that, that's called a vascular bundle. Okay, so again, you got to be able to see those structures, be able to identify them, and if one of them is circled like that, then you know that that's a vascular bundle. Okay, all right. The next one, we looked at the root, and this one is, if you look closely, you can kind of see like a star in the middle, right? And now go ahead and add a line to it. You can see it here. It's kind of like that, right? And that is your xylem. So that's the big empty cells that we talked about. They're non-living. And then around it, okay, these area right here, all these area right here, that's your phloem, right? So that's your phloem. Phloem and xylem, remember, that's a vascular bundle. But in this case, we separated, and you can see the xylem and the phloem very clearly. On the next one is that monkey face that I've been talking about. Okay, the eyes are the xylem, they're empty, and then the brains, that's your phloem. Okay, that's the phloem of the plant where that glucose is actually moving. So you can see very clearly where the xylem and the phloem are located in this cell. Okay. And the last one that we looked at was this one is another stem, and in this one I actually circled the vascular bundle, and you can see the xylem and the phloem. Again, make sure you watch this video again if you're still not familiar with how these structures look. Okay, this is how they look under the microscope. You're responsible for identifying these parts under the microscope, so make sure that you're comfortable at looking at these pictures and identifying them. Okay, all right.